everyone. So today we will be looking at the last lecture for in vivo and in vitro models, where we will look at HIV and HPV models. Okay. Right. So the two viral diseases that we will be looking at is the HIV and HPV model. So we'll start with HIV. So first of all, we will need to understand the pathogenesis of the HIV life cycle. We will first start off with how the viral particles attach to the cell. So HIV is a type of retroviruses where they contain the mRNA nucleic acid material, and they contain an envelope protein encapsulating the nucleic acid. So as you can see, they have a viral envelope here with protein spikes, and these are attaching to the immune cell receptors, so which consists of CD4, CCR5, or CXCR4. So once they are attached to the immune cell receptors, what would happen is it will fuse with the plasma membrane and uncoat, where they will release the internal materials, including viral nucleic acids and viral proteins, where it will assist them to reverse transcribe. So as I've mentioned earlier, HIV is a type of mRNA virus. So they will first undergo reverse transcription before they are imported through the nuclear membrane into the nucleus, where they will integrate into the host genome. From there, it will be transcribed, the viral DNA will be transcribed, translated, where they will be assembled into a new viral particles again. So this assembly process will involve budding and maturation. So there are a lot of boxes here, as you can see. These are all the therapeutic treatments used to inhibit HIV replication. All right. Although there are no effective treatments so far, so these are some of the therapies available, although they cannot fully control its HIV replication. So we have some of the therapy which are used for attachment inhibitors. So it will inhibit the attachment of virus onto the surface of the immune cell. So because this is the first step in order for them to establish infection, so they have developed some attachment inhibitors. Some of them are also the inhibitors for the receptors, for them to bind to the virus particles. Some of them are fusion inhibitors, where it will inhibit the fusion process and the uncoating process. And some of the most famously developed drugs would be the inhibitors that inhibit reverse transcription, inhibiting integration, all right, as well as the maturation inhibitors and the protease inhibitor. All right, so this diagram here summarizes the life cycle of HIV in terms of how they have established an infection by colonizing the cells as a host for them to produce more viral particles. All right. So this is the recap of the stages of the HIV life cycle, where in the step 1, they would enter the target cell via the receptors, where they have fused and uncoat to release the viral RNA to be reverse transcribed, and it will integrate into the into the host genome, where they will produce the viral DNA, where they will transcribe and translate it into the viral proteins, and they will be assembled into the new viral particles again. Okay, so this is the recap or summary of the HIV life cycle. So we have two viruses that causes AIDS, HIV-1 and HIV-2. Both of them belong to a group of retroviruses, which are known to be endemic to the African apes and oval monkeys, and they are collectively known as the primate lentiviruses. So they are primarily spread by heterosexual transmission, and they replicate in the CD4 positive T cells and macrophages. So the cellular proteins that support viral replication includes the receptors, as we have mentioned earlier. Because of these receptors, that they are able to bind to the host cells and subsequently enter into the immune cells. So this involves the CD4 positive or either the CC chemokine receptor 5 or the CXC chemokine receptor 4. Right, all these are the receptors capable of recognizing the HIV virus. And some molecular factors involved include transcription factors, nuclear export factor, as well as the proteins that mediate the maturation, budding of the viral particles, which belongs to the endosomal sorting complex required for the transport. So what are the animal models used for HIV research? The most common model would be the humanized mice. So remember in our previous lecture about the cancer mouse model, so we have also mentioned about how we can generate humanized mice. So it is basically to reconstitute human immune system into the immunocompromised mouse. So basically, you will need to generate a mouse which has human immune system components in it. So in order for a mouse to accept tissues from another species, they will need to be immunocompromised, meaning that these mouse will not reject tissues from another species. So this is how they have been able to generate the humanized mice. So we look at some of the examples here, which they are used as an animal model for HIV research. So we have three here with very complicated names. So what do they refer to? So I will bring you to another article which I've included in the reading materials for topic 2. This is the animal models for HIV/AIDS research. So in order for us to better understand these models, we will look at a journal article. Okay, so let us go to the indicated section. So for small animal models, we have mice, rats, rabbits, right, which are some typical models that they use. So we are looking into mouse model currently. So what do you mean by SCID, HU, THY, and LIV mice? So basically, it means that they are using severe combined immunodeficiency genetic background mice. SCID refers to severe combined immunodeficiency mouse, okay, where they have a mutation in the gene encoding for DNA-dependent protein kinase catalytic subunit, resulting in the absence of both functional B cells and T cells. That's why they have the name called severe combined. They do not produce any functional B cells or T cells in these animals. And in these animals, they are generated by transplanting human. HU here refers to human fetal thymus and liver cells into the SCID mice. So here it is used for as a general understanding of what they have reconstituted into a mouse which is immunocompetent, immunocompromised. All right. So SCID refers to the methods of generating an immunocompromised mouse, and they are reconstituted, means transplanted, with human fetal thymus and liver cells into these mice. All right. So then let us look at the other one. The other one is a SCID human PBL mice. So what's PBL? PBL refers to peripheral blood lymphocytes from human are injected into these SCID mice intraperitoneally and disseminate mainly by the lymph nodes, spleen, bone marrow, and genital mucosa. So this is another strain of mice where they have implanted human peripheral blood lymphocytes into these immunocompromised mouse. Okay, and then we look at the last one, NOD, SCID, IL-12Rg negative. So what are all these terms refers to? So NOD is a type of mouse where they are known as non-obese diabetic mice, right? They are called the non-obese diabetic mice. Okay, so these mouse are generated by crossing SCID mice with the non-obese diabetic mice. So NOD SCID is a result of a crossing between two different types of mice, which in effect, they have multiple genetic deficiencies that result in low host natural killer cell activity, meaning that they have very poor immune defense mechanism. So why are they generating these mice? Why SCID alone is not enough is because they want to improve the efficiency of engraftment with human hematopoietic tissues. Because as you can see from here, they are only using these mice and it is not sufficient to generate a very good model. So hence they have come up with the idea of crossing two different mice line together to generate a more improved model where they are able to increase the efficiency for engraftment with human hematopoietic tissue. So these mice that have multiple genetic deficiencies that result in low host natural killer cell activity and impaired complement activation. So these mouse, okay, however, these mouse, NOD-SID mouse, they suffer from high incidence of thymic lymphomas, which limits their lifespan, right? So this is not something that you want. Mouse with limited lifespan, you cannot test very much in terms of how HIV infections will occur later on. So what they have done is that to further improve engraftment efficiency, they have crossed these mice with another type of mice which is harboring a mutation in interleukin 2 receptor common gamma chain. Hence, you have the IL-2 RG. RG here refers to receptor common gamma chain. Gamma here is a G, receptor common gamma chain. So this is a mice, whereas the protein, this receptor is a protein that is essential for immune cell growth and development. So by crossing these mice, then you have these long term of name, okay, where these are used for HIV research. So what do they do with these mice is that these mice eventually they will be infected by direct injection of the HIV virus into the mouse, which leads to CD4 T cell depletion. So when virus replicates in the host cell, so HIV would always replicate in the CD4 positive T cells or macrophages. So when they replicate in these cells, host immune cells, that results in cell depletion because when they replicate and release the viral particles, it will break the host cell, all right, which results in cell depletion and high viral loads in human tissue. So these mice are generated, okay, immunocompromised, reconstituted with human immune cell system components, and they are injected with the HIV virus, which generate and develop these HIV models for research, right? So what about the other models? We have also the non-human primate models. So these non-human primate models include monkeys, apes, all right? So many species of African monkeys and apes are natural hosts. However, they do not develop disease as a consequence of the infection. So what happens here is although these non-human primates, they are host to a simian immunodeficiency virus, a very close relative of HIV, but they do not develop a disease. In contrast, infection of a special breed of these non-human primate models called Asian macaques, okay, which are not natural hosts for primate lentiviruses, when they are infected with these certain strains of the simian immunodeficiency virus, that results in high viral loads, progressive CD4 positive cell depletion, and opportunistic infection. So in fact, the researchers found that when these Asian macaques are infected with a closely related strain of HIV. It is called SIV, simian immunodeficiency virus. It results in the characteristics which are very, very similar to what we see in HIV patients in human. So hence, they have developed these as a non-human primate model by using a closely related HIV strain called the SIV. Okay, so natural SIV hosts, there are more than 40 different African primate species which are endemically infected with their own unique strain of the SIV. All right, while infection of the non-natural SIV virus, Asian macaques with these strains which are closely resembling HIV that generate these symptoms which are very closely related to HIV, and hence they are widely accepted models. So let us look at some species of macaques which are commonly used for AIDS research and their geographical range. Right, we have rhesus macaque, which are commonly found in the northern part of, or the southern part of China here. All right, and we also have the pigtail macaque. All right, and also the cynomolgus macaques here found in the Southeast Asian region. All right, so this table is important because it allows comparison of the three most widely used animal models for HIV cure research, along with their pros and cons. So we have non-human primate model, murine model, and feline model. So as you can see here, as you compare across the characteristics which used to define how best these models are working. So in terms of sample size, you need quite a number of non-human primates. All right, they have 3 positive here compared to the murine and feline models. And in terms of the anatomy compared to humans, definitely the non-human primate would have an advantage where they are very similar, and mouse and feline models, they are very different. So similarity of virus to HIV. So as we have mentioned earlier, the non-human primate model, they are infected with SIV. It is not the same with HIV. However, for murine model, it is the same virus. And for feline, it is a different virus altogether. It's called the FIV. Okay, so infection characteristics compared to human HIV. So we can see that both non-human primate and the murine model have very good similarities. Okay, hence they are used very widely. So availability for experimental infection in controlled conditions. So yes, all of them. Ability to deplete arms of immune system. Yes. Outbred. Okay, so what do you mean by outbred? So for usually for mouse, they are inbred because they can be used for crossing within the same uh, species. All right. For non-human primate and feline, they are outbred. So in fact, the outbred, right, is a double-edged sword. It can be used to reflect the genetic diversity of samples, but it can also compromise in terms of the complications of how you would be able to um, consolidate your findings. So it is a double-edged sword for this outbred characteristic, because when you have outbred, it it tends to show good relationship in the true environment. However, it will complicate your findings. Okay, so for major surgery required to generate the model, no. Okay, you just need to infect the model. For this murine model, yes, because you will need to generate the humanized mouse, where you will need to first do some crossing for a transgenic mouse for you to obtain the immunocompromised, and then you will need to do transplantation to implant these human immune cells into the mouse itself. So you do require major surgery for you to generate the model. Development of graft versus host disease, no. Okay, yes, for the murine model. Reservoir comparison to human HIV is very similar. Okay, cost to maintain. So of course, your non-human primate model is definitely very difficult to obtain and to maintain compared to the other models. All right. So what are the current limitations and barrier for development of a animal model for HIV research? So this is a very important question for you to address, and okay, we always look at the journal article to help us in understanding what are the current limitations. All right, so we looked at the component here. So for small animal models, okay, like mice, rabbits, cats, they offer obvious advantage in terms of high reproductive rates, low maintenance costs, and the ability to conduct studies using inbred, genetically identical animals. Okay. However, you need to recognize that these species are distantly related to humans. Yeah. So the limitations have been overcome by advances in the development of transgenic mice. Because of these transgenic mice, you are able to study human immune system in a mouse model. Okay, right? But as we mentioned earlier, these humanized mouse model they cannot be bred, meaning that when you have generated an SCID human liver timer mouse, you cannot cross these animals to get the same animal again. Okay, you will need to perform surgery to generate these humanized over and over again for your research, and they have very high maintenance costs. And also, humanized mice are not yet widely used, okay, and accepted for a testing platform for new therapeutics. It is mainly used to understand about the disease pathogenesis. However, for non-human primates, they have very important advantages, okay, because they have over the small animal models, right? The SIV they are used, right? The cell types are susceptible to viral infection, progressive depletion of the CD4 positive cells, and development of opportunistic infection. So, in fact, for the non-human primate model, as we have mentioned earlier, they have very similar characteristics and symptoms to the human HIV uh, characteristics. So it provides an opportunity to test for vaccines and treatments. Okay. And many of the macaque genes controlling the immune system response to HIV are very similar, okay, to the human genes controlling immune response to HIV. So as you can see here, the yellow colored ones here are all the advantages that you are using for non-human primates. So what about the limitations of using non-human primates is that, yes, we have mentioned earlier, outbred. So this will exhibit considerable animal-to-animal variation in the outcome of the infection. However, if I have mentioned, it is a double-edged sword. On one hand, this genetic variability will show the host pathogen interactions, okay? But on the other hand, the genetic diversity will also greatly complicate your studies when you're using a very small number of animals, especially for non-human primate. And ultimately, the viruses they are different. SIV are somewhat different from HIV, where it is not the same virus. So it somehow also complicates the translation of studies where you can use the vaccine developed from SIV for HIV patients. So these are some of the current limitations for HIV research. So I hope you can really go through the journals for you to understand more. Okay, of course, I have also highlighted the important keywords for you to study. So another model that we are looking at is the human papilloma virus. So for human papilloma virus, it can affect both females and males. Okay, it's not only affecting female. Okay, and of course, for now we have vaccines for HPV. So ladies, if you have not taken any, please do consider taking vaccination for HPV because not all viruses have a vaccine, and if there is vaccine available, I think it is quite important, okay, for ladies to take these. vaccines for protection against the cervical cancer. So HPV mainly replicates in the epithelial cells within the cervix, all right, and not all of the infection would result in a disease. Most of the cases, 90% will heal within 2 years, while in some cases, 0.8% will actually develop into cancer, which is relatively invasive. So this is the cervical cancer that I've mentioned. So what are the models available for HPV? We do have several cell lines which are obtained from cervical cancer patients where they have the expression of HPV 18 and HPV 16. So you can use HeLa cells or the other two cell lines here for you to test for as a model for HPV. In some cases, they will also use organotypic cultures. This is the term we use to describe 3D cultures. Right, so these are proliferating keratinocytes cultured on dermal equivalent support. So these can be used to mimic the HPV-induced squamous lesions. Okay, we also have in vivo models for HPV in rabbits, dogs, and bovine, and they are naturally or experimentally infected with different types of papillomavirus strains. Okay, their use as models is very limited by ethical and financial age. So, which is why you understand it's not easy for you to get ethical clearance for models like rabbits, dogs, or bovines. Very difficult. So these are the lists of in vivo models for HPV. So you can see from the list here, these are conventional models, okay, where they are, or most of them are all of them are the mice model. So we look at one of the most commonly used model, which is the K14 HPV16 transgenic mice. So this is a mouse model used to mimic carcinogenesis and immune modulation upon the HIV infection. So these transgenic mice, they express HPV early region genes, including E6 and E7 oncogenes, under the control of human keratin 14 promoter. Hence, you have the name K14 HPV16. K14 is the promoter or enhancer region, which controls the expression of HPV16. So a landmark study in this model demonstrated that chronic estrogen administration promotes malignant transformation of squamous epithelium in the cervix and vagina. So this study here shows the relationship between hormones and the malignant transformation. Okay, so please go through the models for both HPV and HIV models, for you to understand more about viral disease model. So what are the important notes that you need to understand for this lecture is that you need to know the examples of the models used for both HIV and HPV. You also need to understand maybe the life cycle of the HIV virus. How do they establish the infections? Okay, and the pros and cons of the different types of models that we have compared in the table. Okay, for the non-human primate macaques model, the murine model, and also the feline model for HIV disease. All right. So I hope you have learned something for in vitro and in vivo models of human diseases for our topic 2. Okay. Thank you. Bye bye.